Hi everyone, I'm Melis Dalbay. Today I'll be presenting some of the key findings from our recent publication in Journal of Cell Sciences, describing a PCD cell line model generated by BMR1 transduction of basal airway epithelial cells. Primary ciliary dyskinesia is a rare genetic disorder which occurs due to immotile cilia. And in the airways, it manifests as entrapment of mucus leading to breathing difficulties and concurrent infections, which may lead to severe lung damage if left untreated. Unfortunately, currently there are no therapies for PCD. In order to investigate the disease mechanisms and develop therapies, in vitro models are required. However, airway cells lose their capacity to self-renew in culture after a few passages. Here we developed a PCD cell line model that can proliferate and differentiate up to 19 passages and displayed PCD characteristics. To establish the model, we have collected airway epithelial cells from PCD donors with variations in the DNH5 gene, which produces a large altered DNA and protein involved in cilia motility, by nasal brushings and bronchoscopy, and cultivated them to be transduced with a lentivirus encoding BMR1 gene. Subsequently, we have differentiated the cells in airway liquid interface cultures, which gave rise to ciliated mucus-producing cells. Here you can see the primary bronchial epithelial cells from PCD donors before and after BMR1 transduction, and that they display self-renewal markers as P63 and cytokeratin 5, as also seen in wild-type NHB cells. Our PCD cell lines retain their impaired cilia motility, as seen in the high-velocity video microscopy recordings above, whilst the wild-type cells displayed proper cilia motility, as you can see below. We then characterized cilia proteins in our PCD cell line model, and we have shown that the DNH5 protein was missing in the cilia of PCD cell line as expected from their genotype, whilst located in the cilia in wild-type NHB cells. Cilia structural proteins such as CCD40 was present in the cilia of both cell types. Finally, we used our model to investigate PCD cell ion transport activity and measured ENaC and CFTR activity, as they are both important regulators of ion transport and airway surface liquid. We report that in PCD cells, ENaC activity was reduced, while CFTR activity was increased compared to that of wild-type and cystic fibrosis bronchial epithelial cells, and that ENaC was located in the cilia of both wild-type and PCD cells at similar levels, implicating that the decreased ENaC activity in PCD cells could be due to impaired cilia motility. If you would like to know more details, please find our manuscript in JCS Cilia and Flagella special issue. Thank you for your attention.